BPC-157. BPC-157, short for Body Protection Compound 157, is a synthetic peptide derived from a naturally occurring protective protein found in human gastric juice. Although small in size, composed of only 15 amino acids, its biological activity is unusually broad. One of the most widely discussed advantages of BPC-157 lies in its ability to accelerate tissue repair. Research suggests it interacts with growth factor signaling pathways that regulate angiogenesis, the process through which new blood vessels form. Increased vascularization allows injured tissues to receive oxygen and nutrients more efficiently, which may speed the healing of tendons, ligaments, and muscle fibers that normally regenerate slowly due to limited blood supply. Another hidden advantage involves the peptide's interaction with nitric oxide pathways. Nitric oxide plays a critical role in blood vessel dilation, cellular communication, and immune response regulation. By modulating nitric oxide activity, BPC-157 may help stabilize vascular function and protect tissues from ischemic damage, injury caused by restricted blood flow. This mechanism is believed to contribute to the peptide's reported ability to reduce inflammation and protect organs during stress. The peptide also appears to influence fibroblast migration. Fibroblasts are specialized cells responsible for producing collagen and extracellular matrix components, which form the structural framework of connective tissues. Enhanced fibroblast activity can strengthen healing ligaments and tendons, potentially restoring biomechanical integrity faster after injury. Another overlooked benefit is its interaction with the gut-brain axis. Because the compound originates from gastric proteins, BPC-157 has shown protective effects within the digestive tract, including the ability to promote healing of intestinal lining damage and reduce inflammation in gastric tissues. This gastrointestinal stability may indirectly support systemic health because the gut plays a major role in immune signaling and nutrient absorption. BPC-157 may also support nerve regeneration. Certain experimental models suggest it stimulates the expression of growth-associated proteins involved in neural repair. By encouraging nerve fiber regrowth and reducing inflammatory damage around neural tissues, the peptide could theoretically assist recovery from nerve injuries. This combination of vascular support, connective tissue regeneration, and neural protection explains why the compound has drawn attention in regenerative medicine discussions. TB500, thymosin beta-4 fragment. TB500 is a synthetic derivative of thymosin beta-4, a naturally occurring peptide present in many tissues throughout the body. Its primary biological role revolves around actin regulation. Actin is one of the most abundant proteins in human cells and forms part of the cytoskeleton, the structural framework that determines cell shape and movement. TB500 influences how actin filaments assemble and disassemble, allowing cells to migrate more effectively during tissue repair. One hidden advantage of this mechanism is enhanced cellular mobility. When tissues are injured, certain cells must move rapidly into the damaged area to begin repair. TB500 supports this migration process, enabling endothelial cells, fibroblasts, and immune cells to reach wounds more quickly. The result may be faster closure of damaged tissue and improved regeneration. Another important function involves angiogenesis. TB500 appears to stimulate the formation of new blood vessels by activating signaling pathways related to vascular endothelial growth factor, VEGF. Increased vascular density improves nutrient delivery and waste removal in injured tissues, which supports long-term structural repair rather than temporary inflammation-based healing. The peptide also demonstrates anti-inflammatory properties. Chronic inflammation can delay tissue regeneration by damaging surrounding cells and disrupting the normal repair cascade. TB500 may help regulate inflammatory cytokines, allowing the body to transition from the inflammatory stage of healing into the remodeling phase more efficiently. A lesser-known advantage involves cardiac tissue protection. Thymosin beta-4 has been studied for its role in heart muscle repair after ischemic injury. By promoting new blood vessel formation and protecting cardiac cells from oxidative stress, the peptide may assist in preserving heart tissue function after damage. Another notable property is its potential to improve flexibility and connective tissue resilience. By supporting the organization of extracellular matrix components and facilitating collagen deposition, TB500 may help restore structural strength to ligaments, tendons, and muscle fascia. This capacity to coordinate cellular migration, vascular growth, and structural remodeling explains why the peptide is often discussed in regenerative contexts. GHK-Cu, copper peptide. GHK-Cu is a naturally occurring copper-binding peptide composed of 3 amino acids: glycine, histidine, and lysine. It was first discovered in human plasma and later identified in saliva and urine. Despite its small size, this peptide acts as a powerful signaling molecule that influences gene expression across multiple biological systems. One of the most prominent advantages of GHK-Cu is its role in tissue remodeling. Studies have shown that it can activate genes involved in collagen production while simultaneously suppressing genes associated with tissue degradation. Collagen is the primary structural protein in skin, tendons, and connective tissues, meaning increased collagen synthesis can strengthen and restore damaged tissues. Another key advantage is its antioxidant activity. Copper ions bound within the peptide structure participate in enzymatic reactions that neutralize reactive oxygen species. These unstable molecules are generated during cellular metabolism and environmental stress and can damage DNA, proteins, and cell membranes. By reducing oxidative stress, GHK-Cu helps maintain cellular stability. GHK-Cu also appears to stimulate stem cell recruitment. When tissues are damaged, stem cells migrate to the injury site to differentiate into specialized repair cells. Research suggests the peptide may enhance this recruitment process, increasing the body's ability to regenerate skin and connective tissue. Another hidden benefit involves nerve growth stimulation. GHK-Cu has been associated with increased expression of nerve growth factor and other neurotrophic signals. These molecules support neuron survival and synaptic communication, potentially contributing to improved neural repair. Additionally, GHK-Cu may regulate inflammation. Instead of completely suppressing immune activity, it appears to modulate inflammatory responses so that the body can eliminate damaged cells while preventing excessive tissue destruction. This balanced immune signaling allows healing processes to proceed efficiently. Because it can influence hundreds of genes involved in repair, antioxidant defense, and cellular regeneration, GHK-Cu functions less like a single-purpose molecule and more like a biological switch that coordinates multiple aspects of tissue maintenance. CJC-1295. CJC-1295 is a synthetic peptide designed to mimic growth hormone-releasing hormone, GHRH, a signaling molecule naturally produced in the hypothalamus. Its primary function is to stimulate the pituitary gland to release growth hormone impulses that resemble the body's natural secretion pattern. One hidden advantage of this mechanism is endocrine amplification. Instead of introducing growth hormone directly into the body, CJC-1295 encourages the body to produce its own hormone. This approach maintains the regulatory feedback loops that normally control hormone balance, allowing growth hormone levels to rise in a more physiologically coordinated manner. Growth hormone itself plays a major role in cellular repair and metabolic regulation. When released, it stimulates the liver to produce insulin-like growth factor 1, IGF-1, a hormone that promotes protein synthesis, tissue growth, and cell regeneration. Increased IGF-1 signaling can accelerate recovery from physical stress by supporting muscle repair and connective tissue maintenance. Another advantage lies in its long half-life. The peptide can remain active in circulation for extended periods due to a modification that binds it to albumin, a protein that transports molecules through the bloodstream. This extended activity allows consistent stimulation of growth hormone pulses without rapid degradation. CJC-1295 may also influence metabolic efficiency. Growth hormone increases lipolysis, the process through which stored fat is broken down into fatty acids that can be used for energy. This metabolic shift may improve energy utilization while preserving lean tissue. Sleep regulation represents another subtle advantage. Growth hormone secretion normally peaks during deep sleep stages. By supporting natural hormone rhythms, CJC-1295 may indirectly reinforce sleep-related recovery processes, which are essential for neurological restoration and immune regulation. Ipamorelin. Ipamorelin is a selective growth hormone secretagogue belonging to a class of peptides that stimulate the ghrelin receptor. Ghrelin, sometimes referred to as the hunger hormone, interacts with receptors in the brain and pituitary gland to regulate appetite and growth hormone release. One of ipamorelin's main advantages is its specificity. Unlike earlier growth hormone secretagogues, which stimulated multiple hormonal pathways, ipamorelin primarily targets growth hormone release without significantly affecting cortisol or prolactin levels. This selectivity allows the peptide to stimulate anabolic processes while minimizing endocrine disruption. Another hidden benefit is its ability to trigger natural growth hormone pulses. The pituitary gland normally releases growth hormone in short bursts throughout the day and night. Ipamorelin mimics this pattern by activating receptors that initiate these pulses rather than forcing constant hormone elevation. Growth hormone released through this pathway can support tissue regeneration, muscle protein synthesis, and bone remodeling. By enhancing these processes, ipamorelin may contribute to improved structural maintenance of musculoskeletal tissues. The peptide may also support metabolic balance. Growth hormone increases the body's ability to mobilize stored fat for energy while encouraging the preservation of lean tissue. This shift in energy metabolism may improve overall metabolic efficiency. Another potential advantage involves recovery from physiological stress. Growth hormone influences immune cell function, tissue repair mechanisms, and cellular turnover. By stimulating endogenous hormone release, ipamorelin may indirectly support these systems during periods of physical strain. Melanotin 2. Melanotin 2 is a synthetic analog of alpha-melanocyte-stimulating hormone, alpha-MSH, a naturally occurring peptide that regulates melanin production in skin cells. Melanin is the pigment responsible for skin color and also plays a protective role against ultraviolet radiation. One advantage of melanocortin signaling is enhanced photoprotection. When melanocytes produce more melanin, the pigment forms a protective barrier that absorbs and disperses ultraviolet radiation before it can damage DNA within skin cells. Increased melanin density therefore reduces the likelihood of UV-induced cellular damage. Another hidden advantage involves appetite regulation. Melanocortin receptors are located in several regions of the brain associated with hunger signaling. Activation of these receptors can reduce appetite by influencing hypothalamic pathways that control energy balance. Melanotin 2 may also influence immune responses in the skin. Melanocortin peptides possess anti-inflammatory properties that help regulate immune activity during environmental stress such as sun exposure. By moderating inflammatory signals, these peptides help prevent excessive tissue damage. Additionally, melanocortin pathways affect vascular and neurological signaling in the skin. These interactions may influence thermoregulation and localize blood flow, helping the body respond to environmental heat and sunlight exposure. The peptide's ability to activate multiple melanocortin receptors highlights how pigmentation, metabolism, and immune regulation can intersect within a single hormonal signaling system. Thymosin alpha-1. Thymosin alpha-1 is a peptide naturally produced by the thymus gland, an immune organ responsible for T-cell maturation. Its primary role is to help regulate immune function by guiding the development and activity of immune cells. One major advantage of thymosin alpha-1 is immune modulation. Rather than simply stimulating immune activity, it appears to balance immune responses by promoting the maturation of T cells while preventing excessive inflammation. This balance allows the immune system to respond effectively to threats without causing unnecessary tissue damage. Another hidden benefit involves antiviral defense. The peptide can enhance the activity of dendritic cells and natural killer cells, which are responsible for detecting and eliminating infected cells. Improved immune surveillance may help the body identify viral threats earlier. Thymosin alpha-1 also influences cytokine signaling, the chemical language used by immune cells to communicate. By adjusting cytokine production, the peptide helps coordinate immune responses so that different immune cells act in a synchronized manner. Another advantage lies in immune memory formation. When the immune system encounters pathogens, certain T cells become memory cells capable of recognizing the same threat in the future. Thymosin alpha-1 supports the maturation of these cells, strengthening long-term immune readiness. Because the thymus gradually shrinks with age, the production of thymic peptides declines over time. Supplementing these signaling molecules may help restore aspects of immune coordination that weaken during aging. Keep learning, keep embracing what makes you unique. Subscribe and explore the science that makes you extraordinary.